Welcome back to the deep dive. Today we're looking at what is, I think, the single biggest bottleneck in modern genetics.
And it's not the sequencing itself.
No, that battle's basically won. I mean, you can get a genome sequenced for the price of a nice dinner out.
The problem now is the interpretation. The so what?
Right. It's the interpretation crisis. We are just drowning in data, and most of it is in this gray zone.
We call it VUS variants of uncertain significance.
We all know the VUS struggle. You get a report back, and it basically just shrugs at you. It sees a typo in the code, but it has no idea if that typo is, a harmless quirk or the root of a horrible disease.
Exactly. And that gray zone is where diagnostic odysseys just go to die. We've gotten so good at finding the letters, but we're still really struggling to read the sentences.
So today, we're breaking down two papers that are, in effect, trying to teach AI how to read those sentences.
Yeah, one is looking for the where, the specific disease type, the phenotype.
And the other is looking for the how bad, the global severity. We're talking about two models, V2P and POP-EVE.
And these are two very different ways to get at the same core problem. It is reducing the noise.
Because right now, the standard tools are just flagging way, way too many variants as pathogenic.
It's a huge issue.
Okay, so let's start with the first one, V2P. That stands for Variant-to-Phenotype.
Right. And the whole idea here is that the binary system we've been using, pathogenic versus benign, it's just too blunt. It's not a very useful instrument for clinical work.
It's a holdover from how the early databases were built, right?
It is. So tools like CAD or polyphen2, they just spit out a single score. It's like a traffic light, red or green.
But biology isn't binary. A variant that causes, say, a slight hearing problem shouldn't be in the same big red bucket as one that causes fatal heart failure.
Exactly. But let me play devil's advocate for a second. If I'm a busy clinician, I just need to know if the thing is broken. Why do I need 24 different scores? Isn't that just adding more complexity?
That's a fair point. It feels like more data overload.
It does, but think about the clinical reality. You have a patient in front of you. They have a very specific set of symptoms, let's say a skeletal dysplasia.
Okay.
If you run a standard analysis, you might get 50 hits flagged as pathogenic. But 49 of them could be linked to, metabolic issues or skin conditions that your patient clearly doesn't have.
So V2P helps you filter. It lets you sort the hits based on the patient's actual symptoms.
Precisely. It uses what's called multitask learning to generate 24 separate scores. One is for general pathogenicity, like the old tools.
And the other 23?
The other 23 map directly to the big top-level categories of the Human Phenotype Ontology.
Yeah.
So you get a specific score for skeletal system, another for nervous system, another for cardiovascular.
I want to dig into that multitask learning bit. Normally you train a model to do just one thing well. How does training it on 23 different body systems at once actually help?
That's the really clever part. It's all about transfer learning. Biology reuses patterns. A genetic sequence that causes a problem in the heart might share, some deep structural features with one that causes a problem in the muscle.
So by training on everything at the same time.
The AI learns the shared underlying rules of brokenness much, much better than if it just looked at one disease in isolation. It's seeing the family resemblance across different tissues.
Then that led to some really surprising findings, didn't it, about where diseases tend to show up?
It did. They looked at gene essentiality, which is basically how much a gene can tolerate being broken before the organism just can't survive.
And this is where that kind of survivorship bias comes into play.
Yes. They found that phenotypes like abnormalities of the ear, the eye, the genitourinary system, they tend to cluster in less essential genes.
While things like cancer neoplasms or nervous system disorders,
Those cluster in the highly essential genes.
which feels backwards at first. You'd think the most important genes would be the most protected.
And they are by natural selection. If you have a really catastrophic mutation in a gene that's essential for, say, building the heart or the brain, that embryo probably doesn't survive.
It's a miscarriage. We never see those patients.
We never see them. So the diseases in essential genes that we do see are the rare survivors, the ones that just barely made it through.
Whereas you can have a completely non-functional gene for hearing, and while that really impacts your quality of life, it doesn't stop you from being born or having kids.
Exactly. So those mutations can build up in the population. The AI was smart enough to pick up on this evolutionary filter.
They also use this feature selection algorithm, Baruda. How does that work?
Think of Baruda like a tournament bracket for your data. You take your real biological features, gene expression, conservation scores, all that.
Right.
And Baruda creates shadow versions of them, which is basically just randomized noise.
Right.
Then it pits the real data against the noise.
So if your real data can't predict the disease any better than random chance, it gets kicked out.
It's a really strict way to just cut the fat. And the results were so specific. For example, when it was trying to predict skin diseases.
Abnormalities of the integument.
Right. The model independently locked onto genes expressed in sun-exposed skin as its top predictor.
So it figured out the connection between skin disease and sun exposure without anyone explicitly telling it that.
It learned the biological context. And that context is why it just blows the old binary tools out of the water. On a real cohort of immune disorder patients, V2P ranked the actual causal variant at a median rank of 2.
Rank 2. That is something a clinician can actually use. What were the old tools?
KD was at 5.5. KPs was all the way down at rank 10.
A huge difference.
It's the difference between solving a case in minutes and maybe spending days chasing down false leads or just missing it entirely.
So V2P gives us the address of the disease. It tells us which body system is under attack.
Yes.
But that doesn't solve the other part of the equation, severity.
Right. Knowing it's a heart variant doesn't tell you if it's a mild arrhythmia or sudden cardiac death. And that brings us to our second paper, and the model called POP-EVE.
And this one goes right after the crying wolf problem. We saw this recently with alpha-missense. It's a great tool, but if you run a healthy person through it, it lights up like a Christmas tree.
It's a calibration problem. Alpha-missense is brilliant at identifying proteins that are, broken in theory, but it doesn't have a good reference point for what broken actually means in a living, breathing human population.
And it ends up predicting that a perfectly healthy person has something like 5 pathogenic variants.
Which is a clinical nightmare. If the AI says everyone is sick, then effectively no one is. It's just signal inflation.
So POP-EV fixes this by mirroring 2 huge data sets that weirdly don't usually talk to each other.
Deep evolutionary history and shallow human population data.
Walk us through the deep part of that first.
The deep part uses models like EVE and ESM1V. It's looking back millions and millions of years. It's asking, has this specific amino acid at this position been conserved from yeast to mice to humans?
And if the answer is yes and you change it?
Evolution suggests that's a very bad idea.
Okay, so that gives you a raw, this is probably bad score. But that's kind of what alpha-missense does too. Where does the calibration come in?
The calibration comes from the UK Biobank and NOME. It's data from 500,000 mostly healthy people. They use a statistical method, a Gaussian process, to basically curve the grade of that raw evolutionary score.
Curving the grade, I like that. So how does that work in practice?
So let's say evolution is screaming, this mutation is catastrophic, deadly.
Okay.
But then the Gaussian process looks over at the UK Biobank and it sees, 500 people walking around with that exact mutation, and they're all perfectly fine.
It forces the score down.
It curves it down to benign. It says, okay, evolution was worried in theory, but reality says it's fine. And the reverse is also true. If evolution says something is bad, and we see 0 humans with that mutation,
the score gets boosted way up to severe.
You got it. It's fact-checking the deep theory against the shallow modern reality.
And that creates a severity spectrum. This is the really big deal here.
It's the first time we've really seen a tool that can reliably distinguish between variants that cause, say, childhood death versus adult death. Most tools just lump them together. POP-EV puts them on a Richter scale.
And the impact on false positives must be huge.
It's drastic. In the healthy UK Biobank cohort, POP-EV finds fewer than 1 severe hit per person, on average. So when this tool flags something as severe, you really, really listen.
Which brings me to the sort of economic argument here, because normally to crack a rare disease case, you need a trio.
Mom, dad, and child. You need to sequence all 3 to find the de novo mutations.
Which is the gold standard, but it's expensive. It can be hard to get everyone. Sometimes insurance just won't cover 3 genomes.
Right. So if you can only sequence the child, a singleton, you're usually left looking at this massive haystack of variants with no good way to filter them.
But POP-EV changes that.
It changes everything.
Yeah.
Because it's so well calibrated, it can prioritize variants in a singleton almost as well as a full trio analysis can. In their test cohort, POP-EV ranked the actual causal variant as the #1 hit in 98% of cases.
Using only the child's data.
Only the child's data. That
Right.
just massively lowers the barrier to entry for diagnosis. You don't need the whole family tree anymore.
It means a child in a rural clinic with no family history data can still get a definitive answer.
It's a huge step in democratizing diagnosis.
Okay, now I want to shift to what I think is the most the most mind-bending part of this whole thing, the black box validation.
Ah, yeah.
These models, especially they're just statistical engines. They don't know physics, they don't know chemistry, they have never
Mhm.
seen a protein.
And yet, somehow, they seem to have rediscovered structural biology just from the
Now,
math.
the ETF1 gene example, talk us through that, because it really drove this home for me.
ETF1 is a critical protein. It's the one that stops the protein production line at the right time. PopEVE flagged two specific variants in it as absolutely catastrophic.
R190C and R68L.
Right. And when the structural biologists mapped where those variants actually sit in the 3D protein structure, they were just stunned.
Where were they?
They were less than 3 angstroms away from the ribosomal RNA backbone. I mean, we're talking atomic distances here. One was in the specific motif that recognizes the stop sign in the genetic code.
And the other?
The other was right near the trigger that actually cuts the finished protein loose.
So the AI, looking only at population data and evolutionary trees, pinpointed the exact atom-level mechanism that breaks the entire machine.
It gets even better. Take the HDAC2 gene. The model flags a variant, M31R. You map it to the structure. It sits right in what's called the foot pocket of the active site. It literally blocks the enzyme from being able to dock with its target.
Wow. And that has massive implications for drug discovery, doesn't it?
It has to. If the model can tell you the problem is right here in this specific pocket, well, that tells a pharmaceutical chemist exactly where to aim a small molecule.
It moves us from this gene is broken to this specific function of the protein is disabled.
That's the first real step towards a therapy. If we know a pocket is blocked, maybe we can design something to unblock it. If a binding site is loose, maybe we can stabilize it.
It makes the black box of AI feel a little more transparent. The scores actually correlate with physical distance. Variants within 5 to 8 angstroms of a binding partner consistently get higher severity scores.
It proves the model isn't just hallucinating. It's detecting the echo of real physical constraints across millions of years of evolution.
So let's bring it all together. We have V2P, which gives us the address, the specific phenotype.
And we have PopEVE, which gives us the Richter scale, the global severity.
And together, they really represent a big shift toward what's called in silico prioritization.
We're getting to the point where the computer model is becoming as reliable as a basic experiment in a wet lab.
Which we need because you can't run a wet lab assay for every single VUS you find. There just aren't enough pipettes in the world.
We're outpacing our ability to do experiments. We have to rely on computation to filter the candidate list down for us.
These tools let us focus our expensive experimental work on the top 1 or 2 candidates instead of just shooting in the dark.
That's just smart optimization.
Okay, so here's a thought to wrap this up on. We've talked about genotype, the raw code. We've talked about phenotype, the result. And we're using evolution and population data to bridge that gap.
But there's a third pillar we haven't touched on.
The environment. The exposome.
Right.
Because you and I could have the exact same pathogenic variant, but if I live in a city with high pollution and you live out in the countryside, or if I eat a high-fat diet and you're vegan,
Yeah.
the outcome could be completely different.
That is absolutely the next frontier. Right now, these models are predicting the penetrance, how likely a disease is, based only on the genetics. But penetrance is so often a function of the environment.
So if we could feed these models high-quality environmental data with the same detail we fed them genomic data,
Then we stop just predicting disease, and we start actually predicting health. We could say, you have a rank 1 severity variant for this heart condition, but if you maintain this specific lifestyle, we can downgrade that severity score to benign.
a personalized forecast, cloudy with a chance of chromatin failure, so maybe take your vitamins.
It still sounds like science fiction, but with V2P and PopEV, we've basically solved for the genomic variable. The environmental variable is next.
It is a fascinating time to be reading the Book of Life, even as we're still decoding the language. Thanks for walking us through the translation.
Always a pleasure.
And thank you for listening. Keep those neurons firing, and we'll see you on the next deep dive.